Hello, everyone. I am delighted to be able to open the next session, which is dedicated to ATMPs. And I am really happy to have an excellent panel of contributors, of panelists. I will start with ladies, of course. First, Tatjana, who is the scientific CEO of the State Scientific Research Institute in Lithuania. Then we have Peter, who is CEO at Verigraft, and Fredrik, who is the Global Director of Cytiva. And some of the panelists have slides, so now I would like to invite Tatjana to make her presentation first. Of course, ladies first, as usually. Tatjana, the floor is yours.
Thank you very much, Dimitar. Thank you for invitation to this session. Unfortunately, I can't see the panelists. I don't see you, but I see only myself. I don't know.
I think that you have. No, that there is a sign that your internet is not very fast, so probably we can try to present your presentation and you can just deliver. Theodora, can you upload the presentation of Tatjana?
It's okay. I think I can share it, but something wrong with my camera.
Ah, okay. Yes. Now we see your presentation.
Okay.
Yes.
Thank you. So, dear colleagues, dear participants, today I will focus on the hospital exemption for ATMPs, and in particular on its implementation in Lithuania.
You should go, exactly, to presentation. Okay, excellent. Yes.
Okay. So just to mention you, hospital exemption rule allows invaluable opportunity to provide patients with access to innovative, potentially life-saving treatments under the certain circumstances. So, criteria were set in ATMP regulation, and I want just to mention you that is preparation on a non-routine basis, preparations according to specific quality standards, used within the same EU member state, use in a hospital, use under the exclusive responsibility of a medical practitioner, and compliance with an individual medical prescription for a custom-made product for individual patients. So criteria for hospital exemption were not defined enough in ATMP regulation, and its a priori presumes miscellaneous interpretation. So briefly about Lithuanian legislation. The main changes in it were made in 2010. So the pharmaceutical law was supplemented by provisions, and the Minister of Health has launched special rules on manufacture of advanced therapy medicinal products for individual patients, which include requirements for quality, safety, traceability, and pharmacovigilance. Just be brief. We have chosen non-quantitative approach. Evaluation is performed case by case, and what is important issue for me that no permission have been revoked due to a large production scale, and this does not encourage manufacturers to do next steps in clinical trials to get marketing authorizations through centralized procedure. So preparations according to specific quality standards, Lithuania chosen GMP-like rule, not exact, strict following GMP rules. So ATMP in Lithuania must be administered in a hospital where they are prescribed. There are no restriction for use in private hospitals. Also, we has a special prescription blank for ATMP and the hospital exemption, and hospital exemption is not limited only to a high and medical use. So also we have no publicly available information about ATMP. Patients can't make sure if offered ATMP has a permission from national competent authority without a special request. I see it as a gray zone. And the current situation that we have two sites in Lithuania. One is private company and one is in a university hospital, and the total number of permission are 6. So I highlighted specific regulatory aspects in Lithuania. The use of embryonic cells and xenogenic cells is prohibited. Gene therapy products are excluded from hospital exemption. Clinical trials are not required to get a permission. And as I mentioned before, hospital exemption is not limited to unmet medical use. Clinical evidence requirements are also not possible. So, and qualification procedure on a national level is not so clear. So, hospital exemption from the one side facilitates access to new therapies, accelerates innovation process, but the current status is that we have a lot of national variations implementing hospital exemptions. We have misuse of exceptional rules. The use of alternative pathways inhibits the use of hospital exemption in clinical practice. We have an unequal competition with commercial ATMP developments through the centralized procedure, and really we have a known amount of ATMP under the hospital exemption across the European Union member states. That is the current situation, and so thank you, and I'm waiting for questions.
Yes, Tatjana, thank you very much to, first of all, to keeping the time and being so nice to present the situation in Lithuania. There will be definitely questions after that. I have prepared some, and there will be definitely also questions from the audience. But now the next person to whom I would like to give the floor is Peter. Peter, the floor is yours.
Thank you, Dimitar, and good morning, everyone. It's a great pleasure to be here. I hope you can hear me well.
Excellent. Yes, we hear you, we see you, we see that you are in good mood, so please go ahead.
Yes, so the sun is shining in Gothenburg today, so everything is nice here. I will then try to share my presentation with you. Sorry. There we are. So can you see it now?
Yes, yes, excellent. It's in presentation mode, so please go ahead.
Yeah, so I will spend a few minutes and tell you a little bit about how it is to develop an ATMP in Sweden and the type of challenges we see and how we can potentially collaborate throughout Europe in the future. So my name is Petter Björquist. I'm the CEO of Verigraft. Verigraft is a biotechnology company. We are located with our headquarters in Gothenburg. We are a classical university spin-off from the Karolinska Institute in Stockholm. As a privately funded biotech company, we have owners. Our owners are from Europe, from Asia, and from America. So we are really a globally owned company. And we have a management team that has been around for a while. So ATMPs, that is the topic of what we are talking about today. So I would like to just kick off by saying that this field almost didn't exist 20 years ago. So it has went from nothing to one of the most promising fields of medicine today. I would like also to give you my personal reflection about what all this is about. I used to have a picture of a lobster in my presentation. Reason is that, as you can see, this lobster has one big claw and a small claw. And as you also may know, if you break or take off a claw from a lobster, a new claw will grow out and be exactly the same size as before. But unfortunately, if we break off an arm of a human being, that arm will not grow out and form a new arm. So we need to help Mother Nature with regenerative medicine. And I would like to warn you a little bit, because these type of pictures look really fancy and like rocket science, but this type of ear, you can see here, a bioprinted ear, is not really any fancy thing. This is a very simple thing that can easily be made today. But more complex regenerative medicine, as you see here, I also want to warn you for another reason. This is something we cannot do today. You see these type of cartoons in the literature today, but unfortunately, before we can, with a bioprinter, print a new heart or a liver or a kidney, that will take, if you ask me, some 20 years or so. But luckily, we have something that we can do today, and I will quickly talk to you about that. So we are not creating new organs in my company, but we are personalizing organs, and we make them clinically useful. So what is the problem we are trying to solve? As you can see, this is surgery, and today surgery is done for all the big organs. If your heart is failing, hopefully you can receive a new heart by transplantation. But a lot of organs will not be transplanted today, like the non-vital organs, like blood vessels, nerves, skin, etc., for a very simple reason. To transplant an organ from another person, we need to erase the immune system of the patient for the rest of his or her life in order to be able to make that transplantation. So we do that when we have a heart or liver, but not when we're talking about non-vital organs. So we have a solution to that. And how do we do that? This is a blood vessel from a donor. As you can see, this donor is a deceased patient. We apply a so-called decellularization process, meaning that we wash away all cells and other things like DNA from this blood vessel, and we end up with a scaffold that can easily be frozen. We apply a second process being our personalization process. What is unique for my company is that we are harvesting only a small volume of peripheral blood from the patient, and we soak this scaffold with blood solution. We are adding some tricks that are our proprietary solution, and in as little time as 1 week, we have reformed this organ with material being cellular components and non-cellular components, and thereby we have personalized this organ. And this organ is now ready to be transplanted without any reaction from the immune system in a patient. So, well, we believe we have solved the problem, and we believe that we have a future, and our future is starting with this particular indication. In the Thai region or groin region, we have in our veins, in the deep veins, a valve or a couple of valves that are functioning, as you can see here. When the muscle pump in the feet are pumping the blood up, the valve will open, but when the gravity will try to push the blood back to the feet again, this valve is closing. Very elegant, very simple in one way, but unfortunately, some 2 million patients in Europe and North America are having insufficient valves. And what is then happening? As you can see, the blood is going down again without being caught by a valve. And unfortunately, these patients are gradually starting to get pain and swelling of the legs. And the most terrible symptom is that gradually these type of nasty ulcers are formed. We have, of course, done our homework here, and that starts typically in the ATMP field with some health economical analysis and reimbursement models, because that is very important for all of us. We need to see that we can produce a product like this and get it reimbursed by the healthcare system. That is something very central in the ATMP field. Here you see our product. It's called PTEV, the personalized tissue engineered vein. Simply a piece 4 to 6 centimeters long, carrying a functional valve that we have personalized, as I described before. So this is from the reality. So this is a PTEV product, and I think you can see, if you look carefully, the valves are opening when the muscle pump is pumping from below and closing again. So this is the product. And what we are doing now is that we are starting in Europe a phase 1/2 clinical trial. And opposite to what Tatjana explained to us very elegantly, we are using the traditional way of taking a drug to the market. So an ATMP in Europe, being a cell therapy, a gene therapy, or a tissue engineered products like ours, is considered to be a drug and is regulatory handled as a drug. So we need to go through clinical trials. We have just now started and have ongoing the first 15 patients with severe chronic venous insufficiency, as you saw. And this is a 1-year trial. First of its kind worldwide, of course, and we hope to change the patients' lives. So the following on to this is that you need phase 2 trials, efficacy trials, and in discussions with the regulatory authorities, we have estimated some 70 to 80 patients in order to take this product to a first market approval in Europe. We have strong collaborators in Sweden, of course, but also in Spain, Lithuania, Tatjana, as you can see, and Switzerland. So we are really working throughout Europe. In the future, we also, of course, would like to go to the FDA in the US, but that's for another discussion. So to the key point here, the ecosystem we need to bring an ATMP like the one I showed you to the market is the following. So we need, of course, at start, some investors that are taking the risk of bringing this product to the market. This is high risk, but may also be high return in the future. We need a manufacturing partner. We are a small company. We have basically the skills to do the tissue engineering in our company, so we need partners. So manufacturing partners being able to GMP produce this ATMP. We need hospitals that are ready to host clinical trials with ATMP. Still, this is not something that you can find everywhere. We need an experienced clinical research organization heading the clinical trial together with us. We also need surgeons because our products, our ATMP is an organ that should be surgically implanted. And we need surgeons that are really open to do this first in man activities. So taking everything together, I think it's very, very important that we collaborate through Europe. I know that today there is rarely all these things can be found in or be identified in one country. So my suggestion at the moment is that we are collaborating so we can find different skills in different countries, and why not collaborate and take advantage of this. So I hope also that the countries we of focus for today, Bulgaria and other SSE countries, are happy to collaborate with us and other companies and, for example, host clinical trials, because I'm absolutely sure that this is the future for medicine in many ways. As you can see, before I hang up, you see that we have been successful in attracting EU funds, and that is something I really recommend everyone to consider, because EU can fund critical activities in development of ATMPs. And as you can see, they have done that many times for us. So very glad we are in Gothenburg, Sweden, as I said, far away from Sofia and other SSE countries, but I still think that we should collaborate and try to take advantage of each other. Thank you very much for your attention. I'm happy to be around the next hour.
Thank you, Peter. I think that this was a very interesting presentation. Bulgaria has a excellent ecosystem for clinical trials. So probably later on or during the next days, we can talk how we can help you find the best partner in Bulgaria. And now it's my pleasure to give the floor to Fredrik, who is the director of Cytiva. Fredrik, the floor is yours.
Thank you so much, Dr. Dimitar. I will share my presentation.
Go ahead, please.
You can see. Yeah, can you see it now?
Yes, excellent. Thank you very much.
Excellent, great. That's great. First of all, I would like to thank you so much for giving me the opportunity to participate in this very interesting seminar and workshop. So I would like to take this a little bit from a technology provider's perspective, since I'm representing my company Cytiva, based here out of the offices in Uppsala at our HQ. We are today a global provider of different type of bioprocessing technologies and services, with the vision to advance and accelerate the development of life-saving therapeutics and interventions. We are about 7,000 associates, as we call our employees, globally, with operations in more than 40 countries today. And I would like to start from the perspective of talking a little bit about the market and the current technology trends in my area, and then set things into perspective how this is important for ATMPs and how we maybe as a provider can be of assistance in terms of translating innovation and technology over to neighboring countries like Bulgaria and SSE, but not only that, for Europe and the world. So taking a step back, looking at that current market dynamics, we see really from a biopharmaceutical perspective a strong consolidation of the pipeline and moves towards in-country, for-country biologics. And this has been heightened interest due to the ongoing pandemic, of course. We also see smaller companies holding to their drug pipeline or the ATMP pipeline longer than in the past. And I think it's important here to support these to drive their innovation further. Also, of course, very much an increasing molecular diversity in the global biopharma pipeline, constituted of the next generation of biologics, but also biosimilars, which are very important. And of course, the news and industry has been dominated with the emergence of the COVID crisis and so forth. But also through that, we have had an increased, I would say, an extreme high interest in new modalities like viral vectors, mRNA and oligos and so forth, which I think is important because they play a very important role in cell and gene therapy as well. But there is a challenge, I think, from a perspective of technology here. Those new, more complex modalities, in order to be produced according to specification and characterized, there's a requirement for new technology and platforms. For example, viral vectors used in gene therapy or vaccines, they are very much susceptible to aggregation and modification during the production stages. Then we also call, as we discussed in an excellent presentation by the two previous speakers here, the increasing personalization of new therapies and there are small patient populations in comparison to some of the previous biologics like antibodies and protein therapeutics. And here, of course, when you're working very personalized with medicine, the scale up to make this efficient, cost efficient is clearly a challenge. Apart from that, I think, it's important to think about analytics as well in the process of characterizing and making drugs safe and efficient. That's a big challenge for new modalities as well. And I would like to talk about that, mention a little bit more about that in the next few slides. So addressing that complexity with the new modalities in cell and gene therapy, well, when it comes to the vectors that we're using, like adenovirus or adeno-associated virus production, here we need to be able to demonstrate scalability, and we need to have more efficient, cost-efficient processes in order to increase access in Europe, globally to this type of important vectors for cell gene therapy, but also vaccines. There are things ongoing in the cell culture media development for more customized cell culture for these type of applications. And of course, we need to have better purification processes, better chromatography resins for that matter, in order to support the development of plasmids. Plasmids is a starting point for many type of these new type of therapeutics and interventions. And we also need new platforms specific for viral vectors in order to be efficient. So new protocols are needed. And I think from a technology perspective, what we are looking at from manufacturing and quality control is to use smart process development with new statistical approaches, design of experiment, quality by design, high throughput process development, combined with new predictive tools like in silico modeling in order to have better starting points and accelerate development and access to new therapeutics. And apart from that, further development of control strategies to more quickly characterize and quality control new therapeutic products. Another important thing when it comes to, when you've gone through the R&D and the PD, the process development steps, then you need to scale it to a manufacturing scale. And maybe the traditional way you're making factories, stainless steel, is not always the best way forward when you're talking about rapid response to pandemics or addressing smaller patient populations or high risk applications for that matter. Maybe more modular manufacturing solutions, portable solutions is the way forward here for optimized manufacturing and reducing the capital risks of setting up new shop in other parts of the world. So from my perspective, I think technology innovation and some of those new modern tools I mentioned can save time to market, TTM, and drive down the cost of manufacturing. I think another important thing in order to kind of, from a knowledge-based perspective, to outsource this to the rest of the world is standardization and modularization, so you can build and scale as you go, and lower that capital cost burden to set up new manufacturing facilities. And of course also training and resources is a key concern as well, which needs to be addressed. And I'm not going to mention so much about analytical tools and market trends, more than, I think looking at new type of more information-rich analytics is really important. Platform analytical processes, not only platform manufacturing processes, but tools that are multi-purpose, tools that can be used from research through development into manufacturing QC and meet the regulatory requirements. Such new type of analytical tools, I think, are very important for addressing. To the left, you can see here a couple of different things that are in focus today at the biopharma industry. And a tool that can make the handshake between development and manufacturing, that you don't have to do tech transfer between different analytics, is very important to accelerate and drive, for example, the quality and control time frame down 55%, as you can do with some of the new tools for flu vaccines, for example. And I think especially now for more advanced complex biologics and modalities like cell and gene therapy, there's a big question mark, which are the best tools still to be used, because there are other questions that need to be answered there. So we're talking about the viral vector production for clinical use. There are a couple of key challenges. Of course, in terms of setting up the upstream, the cell line part, the purification process, from the transfection to the expression to the purification, as mentioned, here there's a need for innovation to have more efficient processes, better affinity resins for the isolation of the antigen of interest and so forth. But then on the characterization side, I think it's very important as well to look at the analytical part and see how we can move from more labor-intensive, low-precision techniques like qPCR, classical immunoassays that are endpoints that can take hours to get the results, to more real-time detection that can give you more information on how a target is being interrogated on a molecular level, like surface plasma resonance, SPR biosensors, which I think, because of its real-time detection capability, was a great promise at line to characterize, let's say, different subtypes of viral vectors used in vaccine manufacturing or gene therapy vectors. but also to look at maturation of receptors on cells used for cell therapy, and as well as in the future maybe as process analytical technologies that can be integrated in the manufacturing operation to really speed time to market for critical products. So with that, I'm going to pause there, and be happy to discuss throughout the remaining hour. Thank you very much.
Thank you so much. This was really a very interesting presentation and extremely useful as well, because ATMP seemed to be a consolidated expression, but apparently there are a lot of different types of ATMPs, and it was amazing to see what Cytiva is doing. Now we start the questions, and the first question which I have from my end is to Tatjana. Tatjana, it was very nice to see what is going on in Lithuania, in your country, but what is the general situation in Europe with hospital exemptions? Because I don't know a hospital in Bulgaria which is doing the hospital exemption procedure. So what do you know about Europe?
In Europe, really, situation is very different because in this hospital exemption clause, there are some variable and non-variable provisions. And so what concerns variable provision, the diversity is quite huge. So European Medicine Agency has already detected all these issues, and there are some recommendations for future implementation. And first of all, we need to harmonize the conditions and requirements for hospital exemption among the countries. Of course, we need a transparency. As I mentioned, in Lithuania, there are no publicly available information about hospital exemption in Lithuania, but we can see in such countries as Sweden, Belgium, Germany, all the list of available products are on the sites. Even Sweden have a camp, if I'm not mistaken, so a huge amount of information is on sites. So what we need more is appropriate use. When we have no information publicly available, so we have on the other hand, the patient aren't safe with possible and regulated treatments. So it is also mentioned in European Medicine Agency documents. And so they are going to clarifications with all the derogation from directive and regulation. So the huge picture is huge diversity in this field. So basically this one.
Yes, I have another question. Sorry for my ignorance, but
Okay.
if a patient from Bulgaria would like to be treated in Lithuania via a hospital exemption procedure, is this possible?
Now it's not. Okay.
It's not possible.
It's not possible because the hospital exemption is within the country.
So, but my understanding is that European Union is a union where there is a free movement of
that was
and there are also a lot of transborder healthcare initiatives. So, are you sure that this is correct?
Yes, this hospital exemption rule is only for the use within the same EU member states. So the prescription and manufacture must be fulfilled in one country.
Okay, this was interesting to know because this is actually an exemption of the major European principles. But anyway, that's something which needs to be, I think, properly addressed, because when we speak about patient centricity, That's a very nice procedure for the patients in the country, but it does not correspond to the basic European principles. Anyway, thank you. This was very interesting. Peter, the next question is for you. And it was a very nice presentation, but do you have any competing companies who are doing the same like you? And if they are competing, how far they are in their clinical development?
Well, if you start a little bit with the helicopter view, there are many companies and labs and researchers around the world that are following the same type of procedure as we are doing. However, the classical way of doing the reconditioning process is to work with isolated populations of cells. To give you one example, it's very popular to isolate stem cells, and stem cells can, for example, be isolated from the bone marrow. So you take a bone marrow aspirate, you identify the cells you need for your treatment. You have to expand them in vitro in order to have enough cells. And all this is typically a 3-4 months process, maybe up to 6 months process. And that makes a huge limitation for this field, because in order to culture cells for many months in vitro is very expensive. And the regulatory authorities, they are very reluctant, because culturing cells in vitro is a sign of a big risk, because you can easily introduce the risk for cancer formation and all that. So we are not doing anything of that. We are taking blood and we take a small sample of peripheral blood, and we mix that with some enhancing agents, we call them. And then we soak our anonymous scaffold, the decellularized scaffold, for only 1 week. And that makes our process very much cheaper than others. And regulatory-wise, it's much easier for the regulatory authorities to accept it. So I would say, yes, we have competitors, but no, we have no one that are using this type of cheap and simple process as we are doing, because that is really a limitation in the field of ATMPs. The costs for production and the regulatory procedures, they are two of the main challenges. So yes, others are doing similar things, but no one else is doing what we are doing with peripheral blood.
Thank you. This was an excellent question. Actually, 10 years ago, I was owner of one of the cord blood stem cell banks in Bulgaria. Mm-hmm. And one of the projects was also directed in this hypothesis to use scaffolds and make them viable with cord blood stem cells, which of course failed at that time, because it was very complicated first to expand the number of the cord blood stem cells, and also to be sure that there will be no mishappenings in direction of cancer, what you explained. But if it then comes to reimbursement, would this product will be reimbursed as a therapy or as a medical device?
Yeah, it's clear that authorities in Europe is consider a product like the one we have as a drug. So it is an ATMP, not a medical device. And the reason is simple. As soon as you are manipulating cells in one way or another, and that we are doing, because in the blood we have cells that are being seeded onto the scaffold. And if you do that, it is a drug, i.e. an ATMP.
Thank you very much. Now the next question is to Fredrik. Very impressive presentation once again. Apparently you know a lot about the landscape for cell therapies in the world. Could you tell us more something about this landscape, adding on the answer of Peter? How does the landscape perform? Are there any interesting developments? You started with this in your presentation, but would you please explain more? Because it's interesting. Actually, if we would like to create an ecosystem, we should know what are the developments in the world.
I think that's a great question. If we're looking at the global landscape, we have, give or take, more than 1,000 regenerative medicine clinical trials, underway. This is quite interesting because, how are we going to manage the successes in terms of the manufacturing capacity and how are we going to scale things? Looking there are about 40%, I think, 40% ongoing trials within, cell therapy oncology. Just to give that as an example, it's a big focus area, of course, and they are expected to give significant improvements to current cancer treatments and deficiencies that we see for many different type of cancer forms out there. When it comes to the gene therapy space, that's really even I think broader in a scalable manufacturing capacity issue, because here we are talking about not only for the direct gene therapy or DNA type of therapies, we're talking about vaccines as well, using these type of vectors as vehicles for introducing the antigen in the patient. I mean, with COVID, we saw that, and we need to be able to be better at, scaling this quickly and making sure that access is given across the globe in a good and economical, reasonable way. So I think we need to think about how we transfer technology from Sweden, from US, from other places within Europe to Bulgaria, to SSC countries, and so forth, and then have the right incubators and tools in order to scale that further and make sure that we can build up incubators where we can produce locally whenever needed to meet the new needs. Because I think access to ATMPs and the cell therapy and gene therapy, vaccines for that matter, even biosimilars, even the naked antibodies, even in Europe, it's not to be taken for granted or given. I would say, if you're looking at the maps, if you're looking at the health authorities, what they say about access, there are some spaces in Europe still where it's difficult to get the right treatment at the right time. So I think we have a lot of work to do there.
Thank you. I would like to upgrade this question a little bit. I'm dividing the world in terms of cell therapies into big parts. The first part is the so-called regulated world, like Europe, United States, Canada, Australia, Japan, probably South Korea and Taiwan. Where, on the other hand, you have the unregulated world, where cell therapies are developed and applied in a more liberal way, like, for example, Russia, Ukraine, until recently, Thailand, and so on. Where do you think the most advances are done, in the regulated part or in the unregulated part?
That's a great question. Quite difficult for me to answer, since I'm personally mainly working in the regulated world.
Of course, we all are there.
But I would say, in general, I'm working with the US FDA, the health authorities, both in Korea and Japan, China. I meet them on an annual basis to discuss advances in therapy and approaches from a technical perspective. And where I see there is kind of a gap in general, and I guess this goes both for the regulated and the unregulated part of the world. That is the definition of three critical quality attributes of new modalities, and the final critical quality attributes to use for the quality control purpose. So how can we find better ways to secure the final end product, and which type of tools should we use in the toolbox to do that efficiently? That's where I see there could be, from a regulatory perspective, more guidance, especially and it's ongoing in the US. But I think that more guidance would be needed there. In the unregulated part, it's difficult for me to say exactly how the advances there. There may be some places where they have very skilled professors and scientists, and they're doing things that are you know, not common practice in the regulated world yet. But I don't know to say. So I open up that for my fellow participants and yourself to further elaborate on that.
Peter, what do you think about this, about the regulated and not regulated world in terms of cell therapies?
Yeah, it's a very interesting thing to speculate about and discuss. And I think when you give your question, I'm thinking about medical tourism. And I think that this will be a reality in the future. I don't think that people from Europe will go to whatever, Africa or Far East Asia to receive a treatment that has never been tested. But I do think that people may go there as soon as a treatment is tested enough and maybe even approved in Europe. They may go there because in some countries patent protection is not so clear, so that companies or other organizations can produce an ATMP treatment, for example, much cheaper than in Europe and in North America. So I do think that medical tourism will be a part of the ATMP field in the future, but I think we should educate the patients, educate hospitals, educate healthcare systems around the world, because these type of new treatments are of course risky from the beginning. When they have never been tested, they should be tested very carefully first after many years of animal experiments, and the first in man trials should be very clearly regulated and carefully handled. So I think it's a very important thing you are bringing up here. Besides that, I'm not sure about what the future will bring on this side.
Good answer, Tatjana. We have been speaking about over-regulation for hospital exemption, in my opinion, of course. What do you think about the schisma between regulated and not regulated world in terms of cell therapies?
Okay, thank you for your question. So in terms of ATMP, as you know, in European Union, we have, I can say both of them. So we have a traditional way, authorized way, when you go through preclinical, clinical trials and go to European Medicine Agency to get marketing approval. So they launch also PRIME program for medical use, so you can go through this.
Sorry to interrupt you, Tatjana. Sorry to interrupt you. Yes, we know this, but where do you think the biggest advance will be? The bigger advance will be done in the regulated or in the not regulated world?
What I was speaking, so it just
okay. Sorry to interrupt.
What I was speaking about hospital exemption, it's deviation from this traditional path, and they allowed countries to try to use an improved ATMP treatment to patients, and here is variety. And so I think the future is that this data gathered from hospital exemption, you can just use them and put into the marketing authorization. I think the combination of these two methods for Europe, it's very big advantage.
Good, thank you very much. This was a nice answer. So the next question is, apparently the cell therapies in any form still face technical challenges. Could you please elaborate the three biggest technical challenges in developing a cell therapy? Fredrik, I would like to start with you.
I think it's a very important question, because here from a technical and manufacturing perspective, you know, I'm making the kind of analogy back to the classical bioprocessing operation, and here we should maybe change the way we think about cell therapy compared to biologics like antibodies, because here we maybe shouldn't be scaling up things, we should be scaling out things. We need to find ways in the manufacturing plant to have parallel operation, because we are going to do very personalized things for patients, with taking out cells and modifying them. We have cryopreservation operations, and expansion, and we need to do quality controls, and we put them back into the patient again. So here we need to scale out and work in parallel, and we need to find smart ways of doing this in a modular fashion. So I think the way we are working has to change. That's one thing. Even earlier stages, looking at using, for example, plasmids. Plasmids are used. pDNA are used in many different ways. It's used for vaccines. It's used for gene therapy. It's used for cell therapy as well. So here we shouldn't think antibody, kilogram scale. We should think about, we need to have flexible processes that can be applied very quickly, upscale, downscale, depending on the amount needed for the specific application. So here I think it's important to look at building capacity in terms of scalability, depending on the amount needed. So that's how my take on it, except for what I mentioned previously about the analytical part, which still I think is a big challenge where we need to improve quite a bit. But I think there's already now things we can do with modularization and scale out trains instead of scale up trains. And the way we produce plasmids is also improving quite a bit, even on a technical level, in terms of how we express and purify them. So there are things happening, and I think that's where we need to focus more effort to be even better than we are today.
Thank you. Peter, what do you think?
Yes, the product and the products that my company is developing now is not considered cell therapies, just to be clear about that. They are considered tissue engineered product, the third leg of ATMPs. But I happen to work with cell therapies a couple of years back, when I worked with a stem cell therapy for diabetes. And to answer from that horizon, we faced, of course, quite some technical challenges and difficulties, spanning from very basic things like storage of the cells. When you handle cells, it's very complicated to have a living cell population for treatment. So we, of course, tried to freeze and cryopreserve the cells, and that was, at least at that time, a huge challenge. Another example was the purification of the cells, because all cell populations tend to contain unwanted cells. So you should isolate exactly the same, the cell type that you want to use for your treatment, and get rid of all other cell types. And that is, of course, a challenge for all cell therapies to work with sorting of cells, etc. My third example is when you transplant your cells, when you apply them to the patient, the engraftment itself is a major concern, where cells tend to die or be washed away or leave the body, or etc. So to get a cell therapy exactly to the point where you need the effect, that is a huge challenge, and therefore cell therapy developers tend to, for example, encapsulate the cells so they don't leave or escape the site of transplantation. So that was a few examples from my horizon where we do see challenges in cell therapies.
Thank you.
May I make a brief comment? Just
yes, please go ahead. Yes.
I forgot to mention, one thing that we commonly, that is a common theme in the therapeutic world today, and I think also the automation or automating cell therapy from a manufacturing perspective is another challenge that I would like to highlight as well, except for the kind of isolation, the storage, expansion, the purification, and so forth. So maintaining a control of when scaling cell therapies is not a simple task, I think, and I think many people are underestimating the need of competency and expertise to do that. So that's one thing I I think is important for getting a greater efficiency in cell therapies in the future. I just wanted to make that brief comment.
Great contribution, Tatjana. What do you think? Tatjana?
Yeah, I can't comment much on technical issue because we are a research institute with that in preclinical testing. But what I can say from a global perspective that the developers of cell therapies usually are SME or university hospitals, and they usually don't have GMP conditions. They were expensive, and that is the challenge for developers, as I see in our university hospital and in several companies. So it's very expensive.
Yes, so you are partially answering the previous question that probably in unregulated markets, such therapies are applied more generously. So probably there is more knowledge, but less defense and less security for the patient.
Yeah, because ATMP are the drugs, and for drugs, the manufacturer is very strict.
No, it's not only GMP. I think there are also other quality standards which are not met in different countries, but there are some desperate patients who go there to be treated for different diseases. I don't speak at all about aesthetic use of cell products, but just for therapy of some severe diseases. There is a very interesting question from the audience. Do the panelists see a united European science around ATMPs? And if not, how we can achieve this? Tatjana, please.
Could you please,
Yes.
Could you please repeat the question?
Is there a united European science around ATMPs? And in case not, how we can achieve united European scientific effort?
Scientific effort only in European Medicine Agency, the scientific committee. But
no, I mean, there is the science in Europe regarding ATMPs is extremely fragmented. So do you think that this science should be more structured, the scientific aspects?
I think so.
Good, thank you. Peter, what do you think?
Well, yes, I have some experience from this. I think I mentioned that my company has collaborated with organizations in 4 European countries up to now, in Sweden, of course, in Lithuania, in Switzerland, and in Spain. And we have seen some differences. We have also seen that in other aspects, how the national bodies are looking at the science behind the new ATMP is quite similar. But to give you one example, the preclinical package that we have developed, when we have showed and discussed that with the national bodies in these 4 countries, we have seen differences when, for example, some of these countries, they are demanding us to do large animal testing under GLP, while other authorities have said that is not necessary here. So I guess that from my experience, one can say that there are differences, but of course also similarities.
Thank you. And Fredrik?
Just a brief comment on my side. I think, internally at Cytiva, we are, applying our bioprocessing expertise to cell and gene therapy right now to kind of make sure we are retrofitting things to be more effective for this type of new ATMPs. Externally, I think we don't have I don't see a general forum for this externally, but we do collaborate quite a bit with hospitals like Karolinska Institute and Mayo Clinic in the US and others on these type of topics, and we are trying to transfer our knowledge on how to work with functionally closed systems, how to do electronic data capture and try to automate things and to qualify and train people to be more efficient in this space so the innovation can continue. So we do collaborate quite a bit, and there are of course conference and interest organizations that have useful meetings for transferring knowledge, but I don't see this broad collaborative feeling globally on this yet. But I'm sure there will be more forums for this going forward.
Yes, I think so as well, that the pan-European attitude to ATMP is still extremely fragmented. And this is based not only on the cost, but also on the technology, which from time to time is difficult to be transferred from one place to another, or from a university, even from a university to a spin-off. And my last question is today is, how do you think the advances in technology transfer, how imminent they are? Because we need definitely to make a much more aggressive step, if you want, to transfer the technologies easier from the proprietor to the, let's say, to the parties who will produce and distribute. From my perspective, this is still a big obstacle, but I would like to know, what do you think, Fredrik and the other panelists?
Maybe I can start to comment from my perspective.
Yes.
I think it's important to get good and more efficient industry academic partnerships going in this area, so that we can collaborate on some of the fantastic ideas that the academic sector has, and with support from the government to set up incubators where people can come from university, from institutes to test their ideas, where we can give free of charge access to our new technology. Like we have, one example is the Testa Center we have here in Uppsala in Sweden, where we can have people coming in and gaining access to new product introduction, some things that aren't even on the market yet that we are working on, and they can apply if appropriate, then the new therapeutics or ATMPs and try those in our new kits that we are developing. Making that possible in practice is not always easy, but I think doing that in different geographical locations, having more of those incubators and then forming a network of those, it is something I would like to see happening. That kind of advancing this network with a greater transparency and free of charge access to state-of-the-art equipment to get things going, and then finding funding and investors along the way. Maybe this is a naive dream, but I think that, and also providing training from our side, we can provide a lot of training on the technical side of things, from purification, from upstream, downstream, manufacturing, automation, and things like that. So being more generous with that, I think, is important, especially to fight pandemics, but also to address indications with unmet needs with ATMPs.
That's not if at all. I would say that's very realistic. We have all to overcome the fragmentation in the understanding and also with the science, as we mentioned. Tatjana, what do you think about the tech transfer and the problems which we have, and how we can overcome these troubles?
I completely agree with Fredrik, and so I think we need to more efficient dialogue with regulators, because the European Medicine Agency have all instruments to start the early dialogue with developers. So maybe we don't use these tools enough.
Thank you. Peter, what do you think? That's the last question, actually. After that, I will close the session. So,
yeah, thank you. In order to try to answer this, I think we should all conclude and understand that the field of advanced therapies and ATMPs is a very fantastic field, but a very complicated field. And it's spanned on a broad range of different therapies, gene therapies, cell therapy, and tissue engineered product, as you have seen from my presentation, can even be a production of full organs. So it's hard to say that tech transfer can be done in one certain way, if it's possible or not, but in the future we can say that tech transfer will be a very important step. It will be a challenge for the field because some ATMPs are autologous material coming from the own patient that can rarely be centralized in Europe. I mean, while other, for example, allogeneic cell therapies or allogeneic tissue engineering can maybe be centralized, and we can have one production unit in one country, and the product can be distributed all around Europe. But looking at those therapies that need to be produced locally, the tech transfer is key for the future. And I mean, taking my company as an example, we have tech transferred our technology to, for example, Lithuania and to Spain. And the only way we could do that was to send over our own personnel and our own equipment, because in our production we use a bioreactor that we have developed ourselves that cannot be bought on the general market. So we need to ship personnel and ship bioreactors to the hospitals or to the manufacturing sites all around Europe. So this is a delicate challenge that we have to deal with, but I think that we slowly are learning now how to do that and how we can optimize the processes of knowledge transfer in a good way. So by that I think we are partly on our way, but partly this is one of the key challenges.
Thank you very much. Actually, there is one more question, Peter. It's directly to you, but I would encourage the person who put it to send it in a private way, because they are asking you how do you secure your funding, and I don't think that you will disclose this in front of all of us. So the person who put this question, please contact Peter directly, either via us or via LinkedIn or any other tool you can find. We would be happy to support you in contacting Peter, but that's a question which I think should be discussed in privacy, not in front of the entire audience.